Okay, so we are going to go over classification. This is only the first couple of lessons in this unit, um, but the classification part, because it's in the lab, seems to confuse people, so I want to make sure that we go over that. If you have any questions while we go through it, type them in the chat box. So species of organisms, there are 13 billion known species of organisms. And this is only about 5% of all the organisms that have ever lived. So the other 95% are things that have come and gone extinct. And new organisms are still being discovered and identified. And that 13 billion sounds like a huge number. I mean, it is a huge number, but realize that organisms doesn't mean animals. This is plants, this is animals, it's fungus, it's protists, it's bacteria. So 13 billion. Classification is the arrangement of organisms into orderly groups based on their similarities. And another term for classification is taxonomy. Taxa are the groups that we put them into, and the people that identify and name organisms and put them in these groups are called taxonomists. So classification, we're putting them into groups or taxa, so we also call it taxonomy. And the reason that we want to classify things is because it gives names to organisms that are accurate and that they're uniform. It prevents misnomers, such as some of our common names are not, makes it sound like something they're not really, like a koala bear is not a bear, or a starfish or a jellyfish are not fish. So the names that we give them are based in Latin and they're descriptive words. And we can tell from this picture, all of these scientists are looking at the same organism, but we can see that each of them are calling them a different name because they're calling it what they call it in their language. So using the scientific names, the classification or taxonomic names helps to solve this problem because these are the terms that are used internationally. We all use the same taxonomic classification system. And then this guy is Mephitis. So his genus name is Mephitis and his species name is Mephitis. And that means stinky in Latin or smelly, having a bad odor. So we see the terms are kind of used to describe what they are. So early taxonomists, 2,000 years ago, Aristotle was the very first taxonomist, and he divided all organisms into plants and animals. And we know there are a lot of organisms that don't fit into plants and animals, but this is how he divided it at first. And then he subdivided plants and animals based on where they were found. So there were sea dwellers, land dwellers, air dwellers. So now that we've learned more about different organisms, and especially now that we can actually analyze their DNA, of course, um, we can divide them into groups that aren't plants and animals, and we can get a lot more specific. We can see even how closely related things are. And so we said that we all use the same language. They're based off of Latin or Greek. Our next scientist we're going to look at is Carl Linnaeus, and he uh, lived during the 18th century, and he classified organisms by their structure. And so his system is the system that we still use today. And his uh, birth name was Carl, but he changed his name to Carolus Linnaeus, so it sounds more um, like the scientific name, the genus and the species. So classifying them by their structure and giving them that two-word name. Oh, sorry. Here's a picture. This is his uh, publication that he created whenever he came up with his naming system. It was called Systema Natura. So we credit him as being the father of taxonomy. Even though Aristotle earlier did try to classify things, it was incorrect. So once Carl Linnaeus started classifying things based on their structure, that we were able to get more correct names. This system is called binomial nomenclature. Bi means two, and then nome is name. So it kind of translates into a two-name name. No worries. And those two words are the genus and the species. And we always write them the same way, genus first and species. Genus is always capitalized. Species is always lowercase. And then when we're typing them, we italicize them. And if we're handwriting, since we can't really write in italics, we underline them. So at all those different um, levels of organization. So we talked about last week the different kingdoms that we have. I think we talked about that last week. Plants, animals, protists, fungi, and then bacteria. And when the most specific of the two categories are the genus and the species. So an example, I have another example, sorry. So this is the guidelines. We said it's the genus and the species. They're in Latin or Greek. We italicize them when we type them. We underline them when we write them by hand. And then always we capitalize the genus, and then the species is in all lowercase. So our pale-throated sloth is Bradypus tridactylus. And honestly, I don't know what the Bradypus means, but tridactylus, dactyls are your toes or your fingers, and tri means three, so this is a three-toed sloth. And he is adorable. So with our binomial nomenclature, we can also tell how closely related two things are. So if we look at these three different bears, we have the giant panda, which is Liripoda melanalusa. We have a polar bear, which is Ursus maritimus. And we have a grizzly bear, which is Ursus arctos. Which of these do you think are more closely related? So we can tell by looking at their scientific names. Perfect, exactly right. Our polar bear and our grizzly bear are more closely related. And how did you know that? Good, by the first name. So they're both in the same genus because they're both in genus Ursus, but then they have different species names. Uh, the polar bear Ursus maritimus, and maritime is the sea, which makes sense because polar bears, swim. And then grizzly bear is still Ursus genus, but it's Arctos is the species. So we said we only use the two most specific in the scientific name, the genus and the species, but we have much broader groups. Each of these groups we call a taxon. That's the category that related things are placed. So the hierarchy, our very broadest taxon is called a domain, and there's 3 domains. We have eukarya, archaea, bacteria, and bacteria. And then we have our kingdoms. So in our eukaryotic domain, we have our plants, animals, protists, and fungi. Then we have next a phylum, and it's called a phylum for everything except for plants. For plants, for some reason, it's called a division. And then getting more specific, we break them into a class, then an order, then a family, and then a genus, and then the most specific is the species. And species is only the organisms that can mate to create fertile offspring. So for instance, if we mate a horse and a donkey, what do we get? Does anybody know? That's okay. We get a mule, and mules are sterile. They can't have babies. So that tells us that a horse and a donkey are not in the same species. And how I remember the order of them is I use this mnemonic device. Dumb King Philip came over for grape soda so that I can remember domain, kingdom, phylum, class, order, family, genus, species. Dumb King Philip came over for grape soda. So we're going to look back to our bear for an example. So the domain is that they'd be in eukarya because they're all have eukaryotic cells. Then kingdom, we're looking at our grizzly bear. The kingdom that it's in is in Animalia, and we can see other things that are in kingdom Animalia. There's quite a bit of range. Then the phylum is Chordata. Chordates mean they have a spinal column. We can see there's still a lot. We don't see any, besides a spinal cord, similarities between a bear and a rattlesnake, coral snake, sorry. Then they're in class Mammalia, which means that they give birth to their young. They don't lay eggs. Then the order is Carnivora. What do you think that means? Carnivora. What do you think they eat? Meat. Perfect. So they are all carnivores. They eat meat. Then family is Ursidae, and Ursa is bear. It's like we have the Big Dipper that is Ursa Major. Then the genus is Ursus, And then the species was Ursus arctos. So we can see as we get more to a more specific taxa, the organisms become more and more similar. These ones, there's a lot of range. These ones, a little bit less, a little bit less, a little bit less until we narrow it down all the way to our specific species that we're talking about. And then since the time of Carol Linnaeus, we've actually gotten to where, because of the technology, we can do more than even just look at the structure. We can actually look at the evolutionary relationships, which we call phylogeny, and look at similarities in DNA and RNA. The more closely related organisms are, the more similarities we're going to see. So evolutionary relationships or phylogeny, we call this little structure down here a phylogenetic tree, or their specific name for it is a cladogram. Each branch off of it is called a clade. So scientists use physical traits and evolutionary history to determine how organisms are related and to properly classify them. Species with a common ancestor are grouped together along the taxonomic scale, and then species are less closely related as you move up the scale. So how, if we're just given organisms, we make a character table. And so we'd say, okay, what do all of these organisms have in common? And they all have a, I can't even read what that says. It's so small. Okay, well, none of them have everything in common. Well, but they're all animals. How about that? They're all animals. Then next, what do all of them but one have in common? They all have a vertebral column except for the lancelet. So we put our trait there. So everything above the trait has that trait and everything below it does not. Then next, everything but two of them has jaws. So jaws, everything below jaws. So now that excluded the lamprey. Then the next thing said, okay, for walking legs, that excluded the tuna. An amniotic egg, that excluded the salamander. Hair, that excluded the turtle. So as we go up, things are less closely related. So our lancelet and our leopard are not closely related at all. Our tuna and our leopard are more closely related because they're closer together on the tree. A lamprey is more closely related to a tuna than a lancelet is to a salamander. So this is looking at the evolutionary relationships. And then similarities in DNA and RNA. Organisms that are more closely related to each other on the evolutionary scale have more similarities in their DNA sequences. So scientists use the molecular clock, and this is one of the discussion questions. I put in an alternate because this is a hard one to answer. The molecular clock models the DNA comparisons because it looks at how long organisms have been evolving independently from each other. So they can actually see like when that change in DNA occurred. So they can see the timeline of how long they've been evolving separately. So this would be example of like our molecular clock. They started with a common ancestor. Here's our DNA. And then 25 million years later, we can see that they've started to evolve into different species. So we have some differences in their DNA. For this one, here's the difference. And for this one here, here's the difference from the original. And then another 25 million years later, they can see that there's two differences. So we can see how long they've been evolving separately. So the more similarities, the more closely related. So domain is the broadest taxa. It's the most inclusive. Everything is in one of these three domains. And the three domains are archaea. Archaea means ancient. Those are our ancient bacteria. Bacteria, or this one even specifies it a little more as eubacteria, which means true bacteria. And both of the archaea and the eubacteria are unicellular. So the whole organism is only a single cell, and that cell does not have a nucleus or membrane-bound organelles. And then eukarya is more complex. Some of them still can be unicellular, but the cells have a nucleus and have membrane-bound organelles. And we can kind of see the evolutionary branches off of this that we can see the more simple ones up to more complex. All right, then our next we said is kingdom. We have in bacteria, there's only one kingdom, which is eubacteria. In archaea, there's only one kingdom, which is archaebacteria. And then in eukarya, there are four kingdoms: protista, fungi, plantae, and animalia. Our bacteria, eubacteria and archaebacteria, are prokaryotes. And then protista, fungi, plantae, and animalia are eukaryotes, which means eukaryotes means they have a nucleus. Prokaryote means they do not have a nucleus. Then cell structures. Eukarya has cell walls made of peptidoglycan. Archaebacteria has cell walls without peptidoglycan. Protista have cell walls of cellulose in some of them, and then also some of them have chloroplasts. There's quite a bit of variation within the organisms in that kingdom. Fungi have cell walls made of chitin. Plantae have cell walls made of cellulose, and they contain chloroplasts. Animalia has no cell walls or chloroplasts. And eubacteria and archaebacteria, we could see a lot of similarities. They're both unicellular. They both can be heterotrophic, which means they have to get their energy from something they have to obtain it from an outside source. Or autotroph means they can make their own energy. The difference between eubacteria and archaebacteria, archae means ancient. These are our first organisms, and they can, another name for them is extremophiles. They can live in these environments where nothing else can survive. So that's the reason they're so different. We look at this and they seem so similar, but archaebacteria are so different because they can live in these crazy environments. A couple of examples are methanogens. They can live in an environment where they can use methane, they don't need oxygen. Or halophiles, they can live in super, super salty environments where other things can't survive. And then eubacteria is true bacteria. That's more the bacteria that we are familiar with. And some examples of that is streptococcus, when you get strep throat, or E. coli, which you get which, like a food poisoning. Then looking at our eukaryotes, our protists, most of them are unicellular. but some are multicellular. And then some of the unicellular ones are colonial, so they live in colonies. Protists can be autotrophic or heterotrophic. Some examples are an amoeba, a paramecium, slime molds, and a giant kelp. And there's a really wide range in protists. We actually even break that down into animal-like protists, plant-like protists, and fungus-like protists. Then for kingdom fungi, some are unicellular, but most of them are multicellular. They're all heterotrophic. They're called saprophytes. They absorb nutrients, so they don't eat, like a mushroom absorbs nutrients. Examples are mushrooms and yeasts. Our plants, all plants are multicellular. They're all autotrophic. They make their own energy from sunlight. And examples are mosses, ferns, roses, daisies, palm trees. You can put whatever you want for your examples. And then animals are all multicellular. They're all heterotrophic, which means they all have to get their energy from something they consume. And some examples would be sponges, worms, insects, fish, mammals, sloths, cats, puppies. Let me know, girls, when you're ready to move on. I know there was a lot to write on this one. All right. Okay, a dichotomous key. This is the first part of the lab. You have to use a dichotomous key to identify leaves. So the purpose of a dichotomous key is to identify organisms. It's called dichotomous key because characteristics are given in pairs. And whenever you are using one, you have to read both characteristics, and then it'll tell you either to go to another set of characteristics or it will give you the name of the organism. But you have to start at the top each time. So we're going to do an example together. Okay, I know this is kind of small, but so we have two sharks here. This shark is just identifying the different um, features. So we can see this is, yikes. Let me actually pull this up so that we can see it a little bit bigger. Give me 1 second. Okay, okay, I'm going to share my screen. Okay. Does that make it any easier to read? Or is it still the same size? Ah, sorry. That didn't work. I didn't do anything. Oh, now I just don't know what I'm doing. Uh-oh, what did I do? Okay, so the first picture is showing um, the different things on the shark. So this is um, I'll just put it back on the screen then if you guys can't see it any better, so I can point to things. Okay, so this here is the first dorsal fin. This is the second dorsal fin. This here is the caudal fin. This is the anal fin, pelvic fin, pectoral fin, and then these are gills. And then this is the shark that we're going to identify. So we always have to start at the top. If we had 10 different sharks that we were identifying, we'd have to start at the top of the list every single time. So the first question says body is kite-like if viewed from the top, or body is not kite-like when viewed from the top. And when we have all the sharks, it makes more sense because the ones that are stingrays look like look like kites. So we'd say the body is not kite-like when viewed from the top. So we started at 1B, and now we're going to go, it says to go to 2. Pelvic fin absent or pelvic fin present. So this is the pelvic fin. Does our shark have a pelvic fin? Yes. So, sorry, let me see here. So we're going to go to pelvic fin present, and it says go to 3. 3, 6 gill slits present, or 5 gill slits present. How many gill slits does our shark have? 5. So we're going to do this one. So it says go to 4. Only 1 dorsal fin or 2 dorsal fins. And the dorsal fins are the ones on the top. Does ours have 1 or 2? So, oops, these are the dorsal fins. So here's 1, 2, yeah. So it has 2. So go to 5, mouth at the front of the snout rather than on the underside of the head, or mouth on the underside of the head. What does ours have? Underside, good. So at underside, it says go to 6, head expanded on the side with eyes at the end of the expansion, or head not expanded. Not good. Expanded would be like a hammerhead. So head not expanded. So go to 7. The top half of the caudal fin is exactly the same size and shape as the bottom half, or the top half of the caudal fin is different in size and shape than the bottom half. Remember, the caudal fin is the back one. So is it the same on top and bottom, or is it different on top and bottom? It's different, good. So different says go to 8. The first dorsal fin is very long, almost half the total length of the body, or the dorsal fin is regular length. And the dorsal fin is the one on the top. Long, good. So it's very long. So this one says family Pseudotriachidae. So that's what our shark belongs to. And if we did another one, we'd have to start at the top and go down again. Each one will either tell you your answer, your name, or it'll tell you to go to another one. So then our I forget what it calls it. I can't think of what it calls it in the lab, but it tells you, oh, the identification pathway. So for this one, we would say it's 1B to 2B to 3B to 4B to 5B to 6B to 7B to 8A. And they won't all be this long, and they won't always be B, 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 A. That's just how these ended up. So the one for the lab, you're using leaves, and there's a dichotomous key that you use for it. And that is the end. Do you have any questions? Okay, so to earn your points, you can send me email or link or however you want to do it, your notes for this, and that'll be how you earn your credit. Okay? Okay. And then remember, we have tutoring tomorrow at 4 o'clock. So that's if you need help with the lab. That's usually what I get most often. Or in this one, oh, the exit ticket is if you didn't do the notes. So like most likely the people viewing the recording will do the exit ticket. Since you did the notes, you'll send me those to earn your points. Would you prefer to do the exit ticket? The exit ticket is like graded for accuracy. Okay, let me get it. Okay, so there's the exit ticket. Um, and then, yeah, tutoring tomorrow at 4 if you need help with anything. So if you have questions, stick around and you can ask them. If you do not, you are free to go. Thank you so much for coming.